group that extending this to the broader basin is something that's definitely of interest and desirable over this.
Excellent.
This last,
Thank you very much, Anthony. Greatly appreciate your time and your presentation today. We're going to go ahead and transition to the next speaker, so I'm going to go ahead and put you on mute. Again, if people have other questions for Anthony, please, you can follow up with him. I am going to now introduce Dr. Arjan Stevenson from Michigan State University, and he will be presenting relating coastal algal blooms to rivers, nutrients, watershed land use, and storm events. Thank you very much for being with us today, and you will be able to advance your slides with the arrows here at the bottom of the screen, and there is an arrow here for a pointer to make your mouse and cursor work on the screen. I'm going to go ahead and mute and turn it over to you, Jan. Thank you very much.
Okay, thanks, Kara. Good afternoon, everybody. Thanks, Anthony, for that nice presentation on nutrient loading and getting the hour started. What I want to talk about this afternoon is a project that was funded by the Great Lakes Restoration Initiative out of the Year 1 funding. And the project was awarded to myself and David Hyman and Jacko Chi at Michigan State University, and we had a number of graduate students and postdocs and other faculty members that were involved with the project as well, including Anthony, who gave the last presentation. So the goal of this project was really to understand the relationships between land use, storm events, nutrient loading, and phytoplankton biomass. And of course, we know that, especially after seeing the modeling work by Anthony and Emily and that group, that land use contributes a lot of nutrients, and those nutrients get transported to the coastal zones of the Great Lakes via rivers and streams, and storm events exacerbate that loading and carry the nutrients down the coastal zones where they may result in algal blooms. And we have focused our work in the Great Lakes on a few bays and or the western basin of Lake Erie, where algal blooms are particularly bad, and used rather site-specific models. And what we wanted to do with this project was to come up with a kind of a broader approach to looking at the entire Great Lakes and sources of nutrients and potential impacts of those nutrients on coastal zone phytoplankton blooms, or the suspended algal blooms in the coastal zones of the Great Lakes. So to do this, we really felt like we needed to be able to characterize phytoplankton biomass in the coastal zones at spatial and temporal scales that were relevant to algal blooms. And that means that algal blooms, sometimes we have a bad algae year and sometimes we have a good algae year. In other words, there's a lot of variability between years in algal biomass. There's lots of variability between seasons. And typically our sampling of algae in the Great Lakes is very limited on a temporal basis, that we sample maybe once or twice during the summer across the Great Lakes. So we felt that we needed to come up with a new approach for doing this, and I'll describe that in a minute, by using satellite imagery to assess algal biomass across the Great Lakes. The second thing we needed to do was to characterize phosphorus loads across the Great Lakes and into the coastal zones from the watersheds draining into the Great Lakes. The third thing was then relate those phosphorus loads to phytoplankton biomass, and then finally to look at those relationships and for tipping points or break points in the relationships that we could establish potentially as management goals. So I'm going to go through this process with you. So the way we looked at this world was in a relatively simple model, and I'll start from the left to the right in the sense that Geology and soils very much regulate the kind of land use in an area, as well as proximity to the lake. But geology and soils, especially at a global level, really affect where urban and agricultural land use occur. Those land uses generate nutrients. Storms transport those nutrients through the rivers, basically increasing the movement of the nutrients from the land to the water. And then in the water, as illustrated on the right-hand side of the graph, we have algae that grow in the water. And as nutrient concentrations increase, we get a shift from golden brown algae called diatoms to some green algae, and then eventually to when nutrients are high enough, we have cyanobacterial blooms during summer periods. So we wanted to be able to characterize these relationships, and to do that, we used three different forms of information. One, we used existing data, either from maps or from weather records. The second possibility was to use modeling. So in this case, we used geology, land use, and storm events to predict, based on models, what nutrient concentrations would be. And we developed those models, much like the model that Anthony was talking about, but simpler for the entire Great Lakes region. And then finally, we wanted to then be able to relate nutrients to algal blooms without modeling. We wanted to use a statistical relationship where we would model nutrients and then compare that to basically estimates of algal biomass in the coastal zone that were developed using satellite imagery. So I'll start with basically the two parts of how do we characterize phytoplankton biomass using the satellite images, and also how do we develop these phosphorus loading models to the coastal zones. And the idea was to do this over a 12-year time period. So we did this from 2000 to 2012, which was a period that a specific satellite, the MODIS satellite, was operating in the Great Lakes. And we could look at weekly composite images that were available from the MODIS satellite imagery that allowed us to then kind of infer algal biomass in the lakes. One of the problems that we found, although in some ways it's an opportunity, but we found that we were observing a lot of benthic algal biomass in the littoral zone, or we were certainly suspicious that we were. So we made a rule that we had to be far enough offshore that depth was deep enough that we weren't seeing the benthic algal biomass, so that when we did our analysis of the relationships between nutrient loading and phytoplankton, that we were indeed detecting phytoplankton and not benthic algae. The benthic algae is something I'm not going to talk about today, but we did look at some of the nutrient loading benthic algal relationships as well as the phytoplankton relationships. And then I'm going to talk about characterizing the phosphorus loads. So the way that we developed the algorithms or models to predict chlorophyll from MODIS satellite imagery is we used the chlorophyll data that was collected throughout the Great Lakes and collected basically from 77 stations from the years 2007 to 2009. And then we compared that to, and we actually quantitatively related it to the bands and band ratios of the MODIS satellite images. So the idea is that we're going to look at these different colors, bands that are dominantly blue color, like band 3, or green, band 4. But we also use the infrared bands 15 and 16. So we use basically all the bands and band ratios, which gives us incredible statistical power, great statistical power to try to observe phytoplankton condition in the water. And I won't go into details of the model, but we used a statistical technique called boosted regression tree. And the models worked remarkably well, whereas other existing models did not seem to transfer well from one lake to another or one year to another. This is Linda Nowitzki's doctoral dissertation work that shows that whether we transform or don't transform the data, log transforming the chlorophyll data, that we get very accurate models of chlorophyll based on the MODIS satellite imagery and using these boosted regression tree models. So this is how we measured chlorophyll across the Great Lakes. And we then generated maps for the Great Lakes for the summers between 2000 and 2012. So we have maps like this where you can see that we have, here's the scale for chlorophyll in the Great Lakes, and we have relatively high algal biomasses in the western base of Lake Erie, high algal biomasses in Saginaw Bay, and high algal biomasses here in Green Bay. There's also some high algal biomasses up in Thunder Bay and a few other places. But basically we're finding algae where we expect to find it, and we're not finding algae where we don't expect to find it. For example, out in the middle of Lake Huron and Lake Superior. So we're really, we're very satisfied with how these models are working and their robustness for application across the Great Lakes and from one summer to another. The next step was to estimate the phosphorus loads into the Great Lakes. And so for some reason over on the left-hand side, we don't have a table showing up here. But this table listed all the predictor variables that were used in the model. And the predictor variables were, I've got the table here in front of me so I can describe some of these variables to you. Some of them were related to climate. So precipitation is going to determine how much water is moving down, and also potentially what kind of runoff will occur from different kinds of soils. We did use different kinds of soil permeability as indicators of runoff. We used land use, and we used temperature, climate, and then quite a bit of slope and other kinds of aspects of the watersheds as predictor variables for these models of what would phosphorus loading be, what would be the amount of phosphorus that was being poured into the coastal zones given the land use, the geology, and the weather conditions at that time. So these models are allowing us to, at the same temporal scale as our satellite imagery, to put phosphorus into the coastal zones on a weekly time step. In fact, the models allow us to do it even more frequently than that. But that's actually a next step of our research, looking at finer temporal resolution of these relationships. But so we had a number of these observations, and then working with Anthony and Sherry Martin, related model predictions to actual phosphorus loads that they'd measured. And again, we found relatively good relationships between the amount of phosphorus that was predicted to be loaded into the coastal zones and the measured phosphorus load. And these are largely from Michigan rivers. So applicability across the Great Lakes, it'd be desirable to test these models more completely. But so now we go to the next step. Oh, actually, here's a kind of a summary step of which watersheds in the Great Lakes contribute the most phosphorus. So as we can see, the Maumee Basin contributes the most phosphorus load for a summer. So these are summer averages in kilograms per summer. The Maumee, then the St. Joe, and then number 3 is the Seneca River over here in, going into Lake Ontario. Number 4 is the Saginaw Basin. Number 5 is the Fox. So kind of in a problematic way, we have 3 of the top 5 rivers that are contributing phosphorus to the Great Lakes that are contributing them into small to medium-sized to large embayments within the river, or I'm sorry, within the Great Lakes. So we wanted to develop relationships that kind of excluded these hot spots. We wanted to look more along the coastlines of the rest of the Great Lakes and evaluate the phosphorus, the relationship between phosphorus loads into those zones and algal blooms. So, for example, to pick up some of this contribution up near Duluth or along the coastal zones of Lake Erie. Evaluate those situations more. So to do that, we basically estimated the phosphorus coming in along the coastal zone, and then we also defined a 2-pixel wide, Modis pixel wide band of chlorophyll in along the entire shoreline of all the Great Lakes. And now we're going to compare the amount of phosphorus that's loaded to the amount of chlorophyll that's occurring in the Great Lakes at the time that the phosphorus is being loaded, and actually with a lag time that I'll show you in the next figure. Next figure. So here are 4 figures that illustrate the relationship between chlorophyll and depth of the coastal zone, chlorophyll and the distance from the phosphorus loading point, the river mouth, basically, chlorophyll and the phosphorus load a week before we look at the satellite image, and chlorophyll in the satellite image compared to the phosphorus load at the time of the satellite image. So this is the lag time. The lower left-hand corner, that figure has the lag time, and we can see that the effect of phosphorus load is much greater for the week before, with effects ranging from 4 micrograms per liter to 12, so an 8 microgram per liter effect of phosphorus load from the week before, compared to a 2 or 3 microgram per liter effect of the phosphorus load at the time. So also, I want to, before I go into the details of the phosphorus loading and the significance of these factors, I want to mention that distance from the source did not matter nearly as much as we thought it might. So we're going to be investigating that more completely. And depth turned out to be very important. And the relationships makes common sense that with deeper water, there's less effect of phosphorus loads on the algal blooms. So it's only in the shallower waters, and again, deep enough that we're not seeing benthic algae, but in the shallow waters, we tend to get the most effect of phosphorus loading into the coastal zones. So let's look at these relationships a little bit more and think about tipping points. So if we look at just the chlorophyll to phosphorus relationships and tipping points, we could look at both of these relationships and think that we actually see tipping points. That the lines that are drawn through these patterns, through the data, are lines that are generated by a smoothing function. They are not, so this dip in the lower left-hand corner of the lag time chlorophyll response to phosphorus relationship, this dip is not necessarily statistically significant. I mean, we could imagine that a a straight line would not fit through this very well. But it's likely that an asymptotic relationship does fit this line fairly well, that as we add more and more phosphorus into the coastal zone, we get more and more algae, and then at some point we don't get much more increase in algae. So that's one possibility. And if that was the possibility, then we would probably use a point just before it asymptotes out to say that, well, if we control phosphorus at any level above that, we're not going to get any effect. At phosphorus loading, we're not going to get any effect. So that helps us think about restoration of these high-loading watersheds to a point less than 10,000 kilograms per week. That if we don't reduce loading but more than that, we're not going to see much of effect. If, on the other hand, we have a kind of an S-shaped curve where there is some assimilative capacity in the coastal zone before it responds to increases in phosphorus loading and then saturates out, then we can actually establish a management goal, a management target, down around 3,000 to 4,000 kilograms of phosphorus per week. And that would prevent this accumulation of algal biomass. So I have to say that we are not completely sure about what the statistical relationship is at this lower end. That needs more data to some extent. Typically, this is one of the reasons why we're using so many locations along the coastal zones of the Great Lakes to try to resolve these relationships, and so many years of data, so that we generate this large sample size. We can take into account natural variability to try to resolve the details of these patterns. These patterns simply are not observable when we have variable data like phytoplankton data and nutrient loading data without large sample sizes. So we're not sure where we have tipping points. We surely have a tipping point or a threshold that we could think of at the 10,000 kilograms of phosphorus per year. But we really need to think more about whether we have a tipping point at this lower end of the phosphorus range. So with that, just a few conclusions. That we were very happy with the way that both Landsat and the MODIS imagery were predicting chlorophyll. And the Great Lakes-wide phosphorus models were sufficient that with our chlorophyll estimates, we could develop environmental relationships. We could relate phosphorus and remotely sense chlorophyll concentrations over multiple years and over multiple over all the Great Lakes in a way that made sense, that made ecological sense. And as far as tipping points and thinking about the relationship between phytoplankton biomass and phosphorus loading, the 1 week lag turned out to be an interesting observation that peak biomass occurs 1 week after the phosphorus loading, and 10,000 kilograms seems to be a threshold associated with the phosphorus loading and the offshore constraints in these open water zones that could be related to a tipping point. Whether there are additional tipping points is going to be important to investigate larger sample sizes and do more research. So with that, thank you.
Thank you very much. We really appreciate your presentation today. We do have David Hart just provided some information. David, I'm going to try to go ahead and unmute you. If you'd like to address the information as well here, I'm going to make you a presenter, and then if you want to go ahead and unmute yourself at the top if you'd like to discuss this post that you just put on chat. And if anyone else has a question they would like or a comment they'd like to make, please go ahead and put it into chat, and I will work to unmute you as well. We have just a couple of minutes left, but we can discuss in the time that we do have left. Let's see, is that working for you?
So I can read what David has sent, and we would be really interested in the Fox-Wolf hydrologic dashboard, and actually probably talking to them more about the information that they have. That we're very interested in kind of following up on research on these embayments. And we've done more intensive work in Saginaw Bay looking at the long-term algal biomass nutrient loading relationships using Landsat all the way back to the 1970s. And in that work, we're concerned about temporal resolution, getting daily kind of estimates to see if we were missing peaks in algal blooms. So to test that, we went to Green Bay, and we're using the satellite images and doing daily analyses of phytoplankton in Green Bay and linking that to maybe some of the work that they're doing in the Fox-Wolf River would be desirable. So, David, if you'll contact me by email, that would be, and want to follow up that way if we don't get this problem solved, that'd be, if we can't communicate directly here, that'd be great.
And it is 4 o'clock, so it looks like we will need to wrap up today. Thank you, Jan and Anthony, for your presentations today, and for the participants for taking their time to listen in today as well. Our next session will be on Friday, October 18th. Coastal Wetland Biodiversity and Tipping Points will have presentations from Katia Kovalenko and Ed Rutherford. I'll look forward to talking to all of you then, and I'll post the webinar video on our website soon as well and email that out to everyone. Thank you all very much and have a great afternoon.